Welcome to video 3.8. In this video, we'll be covering tertiary structure, specifically disulfide bonds and prosthetic groups. This will wrap up the tertiary structure section, and our last section, quaternary structure, will be next, wrapping up protein structure as a whole. So, looking at disulfide bonds, this is an interesting topic, and it's important to remember that we'll be talking about cysteine here. So when we think about cysteine, or SCYS, this amino acid is ionizable. And specifically, with a pKa of 8.5, that means that at pH 7, we're mostly going to be dealing with neutral, like SH, the HA form, so to speak, of this amino acid side chain. But as we get to higher pHs, we're going to start seeing a lot of negative charge. Right? We're going to start seeing the deprotonation of cysteine into S-. And the important idea is that when S- exists, the two cysteines can react together. So, and that forms a disulfide bond, which I'll show you below. Importantly, where do we see pHs like this? It's actually realistic in a cell, right? We see this in what are called reducing environments or non-reducing environments, right? So certain organelles of the cell, especially for proteins that are being secreted out of the Golgi, which is a little more detailed than you probably need to know. But these are realistic pHs that we can see inside cells. Even if we get close to pH 9, even if we were at pH 8, for example, we'd still have some S-. Now, when it exists, two different cysteines can react together, and you make this covalent bond, the SS bond. Sometimes we call this a cysteine linkage, so be careful, it's not cysteine with an E, it's cysteine, just with the I. That's the name of this linkage. To avoid that confusion, I just like to call them disulfide bonds. Two sulfurs reacting together. This is a very strong linkage, and it's covalent, so it's unlike a hydrogen bond, it's actually a real bond between these two atoms. So why does this matter for the three-dimensional shape of a protein and tertiary structure? We can view disulfide bonds as locking tertiary structures. So here's an example of a polypeptide sequence from N to C. We have 4 different cysteines, white, black, red, blue, and purple. And we can imagine different combinations. So for example, this sulfur is able to make a linkage with this one. That would be the case 1 up here. And then the purple and blue could make a bond as well. That would keep, it kind of makes a new bond that connects these things and forces them to be close together, right? Giving you some sort of structure. In the second case, though, we can see the same thing just across the molecule, right? What's stopping the black cysteine from making a disulfide bond with the purple one, right? As this thing is folding together, if these two sulfurs find each other in close proximity, they could form a cysteine bond, and then the blue and red would form a cysteine bond, and you would have that kind of situation, right? We can also imagine a second, or rather a third case, and in this third case, right, you can see all these different combinations become quite challenging to organize, but the black one could connect to the blue one, and the red one could connect to the purple one. Right, so we have all these different combinations of cysteine bonds that can form, and this plays an important role in many proteins' tertiary structure. Right? Many times proteins will keep folding and refolding until they have the correct arrangement of sulfurs. That's, again, more complicated than we need to dig into, but it's important to recognize the relevance of cysteine, specifically the amino acid, not just as an ionizable side chain that takes on a negative charge, but also the amino acid responsible for disulfide bonds. With that, we have covered most of the different things that impact tertiary structure and the correct folding of a protein. And it's important to note, very briefly, that when it comes to a protein folding, the idea is that there's a low energy state. And as you try to fold, there are many different forms you might end up with, but there's some sort of valley where this is, energy stability on this side. And this is just all the different potential forms you could have. And one of these forms is going to be the most stable. And eventually, as the molecule keeps trying to fold and all the crazy chemical interactions are happening, you'll eventually find yourself at this point of stability where any changes you make to that protein structure make the protein worse. right? It's at this point that you've arrived at the correct fold of your protein. So when I discuss, multiple types of folding, it's a very complicated process. I'm explaining very simply here, but just wanted to get across this idea that most proteins will fold and reach their stable place based on this principle of just finding the most stable conformation. The last thing to talk about with tertiary structure is the use of a prosthetic group. So to kind of make this term make sense, why we call it prosthetic, we can imagine a person here missing a leg, but we can offer a prosthetic leg, right? That prosthetic leg is not part of the person, but it helps them achieve a specific function, right? This is still a human being, but maybe this is a human being that has trouble walking. There's now a function of walking. So this prosthetic group is required for the function. Therefore, we call it an important part of the protein. For hemoglobin, to move it to an actual protein, hemoglobin is the protein in your blood that carries oxygen. Right? It turns out, though, that hemoglobin actually can't bind oxygen. You specifically need a chemical called heme, which we'll talk much more about in section 4. That's the thing that actually binds oxygen. So much like the above case, this hemoglobin protein, it's a protein, but it doesn't have a function. It's not functional. We call this the apoprotein, the incomplete protein. However, when we have the hemoglobin with the correct prosthetic group, heme, which is just a chemical that allows for oxygen to bind, now hemoglobin can achieve its function of being an oxygen transport protein. So this is now called a holoprotein. So again, apoprotein and holoprotein. The important idea here is that, like the prosthetic leg isn't, made of flesh and blood like a regular leg, prosthetic groups are not necessarily protein, but are an important part of the protein's tertiary structure because it's required for function. With that in mind, the only topic we have left to cover is that of quaternary structure. I'll see you in the next video to discuss that.